Welcome friends, welcome to our YouTube channel, and we are studying the chapter that is fertilization. This is the third video lecture on the topic fertilization, and today we will going to study about another process that is called as the acrosome reaction. Till now we have studied regarding the capacitation of sperm and movement of sperm toward the ovum. Okay, so whenever it reaches toward the ovum, it encounters with another layer, and that is called as the cumulus. Okay, and whenever it interacts with the cumulus, it undergoes some sort of reactions. Okay, so there is two cases arises here. Two cases arises. First one is, as per the speed or as per the motility of the sperm, it penetrates the cumulus ovirus and reaches to the another layer that is called as the zona pellucida. Okay, and if it reaches to the another part that is called the zona pellucida, so there could be two fate of this sperm head. First, either the acrosome barrier will be intact, means either the head which is enveloped with the help of the acrosome vesicles is intact, or it may have fused with the cumulus. So there could be the two fate, या तो वो क्या होगा एक्रोसोम हेड से इंटैक्ट होगा, या फिर क्या होगा वो ऑलरेडी क्यूमुलस से वो फ्यूज कर गया होता है. सो हमारे पास दो एस्पेक्ट्स है. ओके, सो वी विल बी डिस्कसिंग दिस थिंग्स लेटर what exactly happens, but before proceeding toward this process, let us understand, okay, let us understand the interaction of the sperm and ovums zona pellucida with each other. Okay, so if you can see the figure here, so its figure can be seen that the zona pellucida, in case of the mammal, there are, in case of the human being, there are four types of the zona pellucida. Zona pellucida 1, 2, 3 and 4, and in rat it is of three types. Right now we have seen here, we have represented only three types of the zona pellucida, that is 1, 2 and 3. So it can be easily seen that here is a sperm here is a sperm head, this is a sperm head. So basically this is a sperm. Okay, and this is the arrangement of the zona pellucida. Just we have tried to make present here the three layer of the zona pellucida which is intact with each other. Now here is ZP3, ZP2. This is an alternate arrangement of the zona pellucida, and there is a representation of the zona pellucida 1. First, we have gone through this structure ZP1, ZP2 and ZP3 arrangement. Now let us understand what are the factors which are just present on all these things. So here is the zona pellucida 3, and this is a carbohydrate molecule, and that is called as N-acetylglucosamine. This is N-acetylglucosamine, N-acetylglucosamine. Okay, and with this N-acetylglucosamine, this is the acrosomes. Let us understand, let us suppose that the head of the sperm, that is called as the acrosome, is intact here. It is not diffused, it is not broken while passing through the cumulus. Let us understand it is intact. Now then this is already we have gone that this is a carbohydrate molecule that is called as the N-acetylglucosamine, and the receptor is made up of this receptor. This receptor is made up of galactosyl transferase. This is the receptor, and this is galactosyl transferase, which is expressed on the outer membrane of the acrosome vesicles. Okay, now let us understand another thing. Whenever the sperm binds with the help of the zona pellucida, okay, with the help of the zona pellucida, and this is it binds to zona pellucida laterally. Suppose this is the head of the sperm, and this will bind to this layer laterally. This will bind to there laterally. So this is the seed molecule, CED1. This is a seed protein, and through which it is bind to the ZP3 and then to ZP. It is bind to ZP3 and then to ZP2. Okay. Now, what happens? What happens here? Whenever the sperm reaches to the zona pellucida membrane, okay, so firstly it needs to be adhered with the surface of the zona pellucida, okay, and this adherence is induced with the help of the galactosyl transferase, which binds to the N-acetylglucosamine, and another bound is the CED1 protein, from which it binds to the zona pellucida molecule laterally, okay. Now let us understand, whenever N-acetylgalactosyl transferase binds only with the ZP3, okay, galactosyl transferase binds only with the help of the ZP3, and ZP1 is the, just you can say it is a layer between the two row. This is the layer between the two row. Now what happens with the help of the motility? Okay, still right now the sperm has come here and bound to the zona pellucida membrane firmly with the help of the galactosyl transferase, which bonds with the help of the N-acetylglucosamine, and another attachment is with here is with the help of the SED1 protein. So now, and with the help of all this, it's just make a bore into the, it makes a bore into the zona pellucida membrane, and it reaches to the another part of the sperm, that is called as the upper part of the ovum. It reaches to the upper part of the upper cell membrane of the ovum. Okay, so after it reaches from here, and by just making a hole or making a pathway through the zona pellucida layer, it come across toward this region. It come across toward this region. So what happens? Okay, what happens here? What happens here? So just you will just you will say that the after it binds with the all these substances which is present on the zona pellucida and on the surface of the sperm acrosome, what happens here? Okay, so acrosomal, there is a burst of the acrosome. Okay, there is a lysis of the acrosome occurs, and whatever the proteins are present over here, already we have discussed different type of the protein in the previous lecture. With the help of the calcium mediated exocytosis of the acrosome mammal occurs, and what happens, the secretions of the acrosome occur, and ultimately what happens, it digests the matrix of the zona pellucida, and by making a hole toward the region, and it reaches to the, now what happens with them, it is mediated with the help of the acrosome reaction, and now it just okay. So this is the acrosome enzymes, and now this sperm moves towards the another region of the sperm of the ovum. Okay, so let us understand the another part of the ovum, and that is called as the fusion of the sperms on the surface of the egg cell membrane. So suppose this is the egg, okay, and also okay, so this is an ovum. Now this is the cell membrane. Now what happens? The sperm comes here after traversing through the zona pellucida. Now the structure of sperm is somewhat like this. Suppose this is a rough diagram. And most of the most of the acrosome enzymes has been dispersed, dispersed, and which helps to pass through the zona pellucida. Right now what happens here, whenever it reaches whenever it reaches in between the cell membrane and the space that is called as the between the cell membrane and the zona pellucida, and we called is a pre-vitelline membrane. So what happens here, ki there is some sort of protein, which is present over the equatorial region of the sperm. So let us understand what is the equatorial region of the sperm. Okay, now suppose this is in a sperm and this is a sperm and this is acrosome. Okay, and this is the inner part, inner part of the acrosome. Okay, and this is the head, outer layer of the head. Okay, and the space which is between the inner part and outer part of the sperm, inner part of the acrosome and outer part of the sperm is called as equatorial region. Okay, equatorial region. So here on the equatorial region, this is suppose equatorial region, there is a protein which is present on the inner part of the acrosome layer, and that is called as the izumo. Izumo is a Japanese serine. इजुमो is a Japanese serine for a marriage. What is serine? एक पवित्र स्थल है जहां पर विवाह होता है. And on the surface, or you can say on the surface outer membrane of the ovum, there is a projection. There is a projection of the villi, just like a villi. There is a projection like a villi. And this projection is called as the zuno. This projection is called as the zuno. Okay, so what happens here, and you will understand what about the zuno. So the zuno is also a protein, and in Roman it is a goddess. In Roman it is a goddess of marriage and fertility. In Roman, it is a this is a goddess of fertility and the marriage. So now we come across the two term, two protein, izumo and the zuno. Izumo is present on the sperm head and zuno is present on the surface of the ovum. Okay. Now what happens? Okay. Izumo and zuno, izumo and zuno combine with each other. Okay, izumo and zuno combine with each other, and what happens? It fuses with the outer membrane of the ovum. It fuses with the outer membrane of the ovum, and remember here there is a myth that there is a hole occurring. So there is no hole occurring on the surface of the ovum with the help of the sperm head. There is no hole occurring in this layer. Okay, so what happens? It fuses with the, after binding, it fuses with the cell membrane of the ovum. Okay, it fuses with the cell membrane of the ovum. So let us understand with another diagram. Suppose this is a layer of ovum where and right now okay, so this is izumo and this one is the zuno. Okay, they interact with each other, and what happens? The membrane of this region will fuse with each other, and remember here there is a packet of, you can say, the cortical granules as there. Now what happens here? Whenever it binds with a sperm head, make a path after fusing. This is okay. So this is a sperm head, and this sperm head make a path after fusing. Remember, there is no hole occurring, but it fuses with the sperm. It fuses with the membrane, and it reaches inside. And it reaches inside the cell membrane, and in this way it reaches, this is the nucleus, and in this way it reaches inside the cell membrane, and meanwhile it also it also fused with the packets of the you can say, and that is called as a corticals granules. Okay, and meanwhile it fuses with the membrane of the cortical granules. Cortical granules comes out. Okay, cortical granules comes out and the sperm comes in. Okay, so this was just an brief idea regarding the interaction of the sperm with surface of the egg membrane or the cell membrane of the ovum and with the zona pellucida. Okay, now let us understand there is another protein, and that is called the CD9 protein, which occurs CD9 protein CD9 protein, which occurs on the surface of the ovum. But what actually it does, it is still a controversial thing. Sometimes it is said that it binds with the Adam 2, which is present on the surface of the sperm, but it is not clear right now, or if it is clear, then till now I don't know. And and there is another question that if there is an absence of the zuno or any of this zuno or izumo, then what will happen? There is a case study. Suppose there is no zuno or izumo protein is there on the surface of the egg, on the surface of the you can say surface of the sperm, then what will happen? So there is nothing. It will traverse towards the zona pellucida, it will traverse toward the cumulus operus, but it could not be able to fertilize with the egg, and it could not travel throughout the you can say the ovum. The same situation is also with the case of the CD9 protein. Whenever there is no expression of the CD9, so there will be no fusion of the sperm with the help of with the egg. And now this was about the acrosome reaction. In the next lecture, we will come to understand about the cortical reaction and the stopping of the polyembryony, or you can say polyspermy. Thank you. If you like the video, please don't forget to share, subscribe and comment. Thank you. Thanks for watching.